All right. First of all, just want to say, after 15 years of leaving the PRL, feels real good to be remembered, and want to thank the committee that invited me here for thinking of me. It means a lot. And when I got the invite a couple months ago, PRL at 60 years, it took me a minute, but what came to me was like, for a great institution like the PRL is, this is a youngish age, right? And before I get into talking about and reflecting on, my years here and how that's impacted my career in the past 15 years since I left, just wanted to say to the graduate students, the postdocs, staff, and faculty here, you guys are the ones who are going to make the discoveries and build the future of this institute for the next 10 years so that we can all come back and celebrate even greater things than we are today. So to that, what I want to say to everyone is onward, right? And obviously that's something to say in a world where we face a lot of challenges, and it's, talk can be cheap. But one of the messages I want to get across today is never say never whenever you're feeling challenges in your work. I felt a lot of mine. I'll talk about navigating some of those through the years, as well as other reflections that I've had since leaving the PRL and leaving academia, for that matter, and going into industry. And just reflecting back on a lot of the things that I learned here at MSU and in the PRL, in Christoph Benning's lab, that really impacted me and really benefited me along the road. So before I get into that, I think I want to rewind and start rolling the tapes. At the beginning of the century, before I joined the PRL, I was an undergraduate student at the University of Nebraska, and maybe by my second year, it just became so clear to me, like, plant biochemistry is it for me. This is it. Found the first lab that was willing to take me in. It was Ray Chalet's lab. He was an excellent protein biochemist that was doing work in soybean root nodules on the PEP carboxylases. But when I got an interview with him, he said, you could work on that if you want, but I just got this grant with Chlamydomonas, a green algae. I'd never heard of it before. And he said, I want you to think about that, because as an undergraduate, you've got your coursework, you got all this stuff that you're going to have to do outside of the lab, and you might find yourself spending most of your time taking care of plants, waiting for 2 root nodules to grow so you can do a single experiment. With Clammy, you can do a couple experiments a week, screw them up, and then finally get things right and make a lot of progress and really learn hardcore protein biochemistry, molecular biology. And so I jumped in on that, did a lot of great work on algae, but was still convinced, working with plants and plant biochemistry is where I want to be at. And getting closer to applying for graduate school, talked with him, he said, one of the places you got to check out is the PRL. It has the highest population density of passionate and excellent plant scientists in the world. You can throw a rock and you're guaranteed to hit someone who's worth learning from and talking to. So I applied for that, and right around the same time, Mike Tomashow came to the university and gave this excellent talk on cold acclimation, the CBF pathway, the cold regulation, Regulon, and this hunt that he and his lab were on to find this temperature sensor. And it was like late November and like 33 degrees outside, and it had been raining. And by the time the talk ended, it had turned into sleet, and anyone who's been in Nebraska knows it's also blowing at 25 miles an hour. And that story just resonated with me, and I was like, I hope I get the invite to go visit the campus. That happened, visited other places, but it was just clear as day after the visit. Coming to the PRL was going to be the right move for me. Joined, got right down to business, rotated through first in Mike Tomashow's lab, and I think at the time we're working on yeast 2 hybrid screens to find some interacting proteins. I don't remember the details, but what ended up happening, and this was kind of a feature that was important for my decision to join the PRL at the time, was that you could rotate through other labs outside of the PRL, and I ended up deciding to join Christoph Benning's lab and work in chloroplast lipid metabolism. At the time, things were real hot and heavy on a couple of different mysteries in chloroplast biogenesis, lipid trafficking, and a few other things. And this was an area of metabolism that I didn't know a lot about, and it was just fascinating to me and really challenging too. The protein biochemistry is really hard to do. The lab had this strong focus and track record of finding these hard-to-find genes with mutant screens. And this was a fascinating and completely different approach. Fell in love with the project. The team, working with a big team versus a very small team of two people with a professor who was about ready to retire, was just an amazing experience for me. It went really fast. And it was fun because it turns out all of the lipids and the genes that encode them, the words are way too long to say. There's way too many of them, right? And so we had a scheme to talk about everything we needed to talk about using only 5 letters of the alphabet, right? MGD1, DGD1, the workhorses. You had TGD1 through 5, the lipid trafficking system that we were still trying to figure out, colleagues were trying to figure out that got the substrate to those workhorse enzymes. There were also like a couple of white whales. There was a big mystery, a suppressor called DGS1 I worked on, never figured that one out. But then the big one that we didn't really have active funding for was this enzyme called, I'm going to try it here, we'll see if I can say it, the galactolipid galactosyl transferase, or GGGT. And this one, I'd had back burner projects on for a couple years. And at the time, my vision was, I thought it was so clear. I'm going to do this. I'm going to find the right postdoc and then find my way into a faculty position, pick the singular color of shirt that I'm going to wear for the rest of my career, and just forge on. But what happened about halfway through is Christoph came out of the lab. And he said, this guy from the Air Force Office of Scientific Research just called me and he said, do you want to apply for a grant for algae biofuels? And he came out and he's like, you used to work on clammy, didn't you? I said, yeah. He's like, should we do this? And I said, let's do it. And within, what was it, like 5 or 6 weeks, the money had landed and we had to scramble to hire a team of people, which included at the time his son, who was in high school, one of his friends, a master's level technician, joined the crew, and starting to work on these fundamental problems of how do lipids accumulate in algae, what are the mechanisms, and it turns out that path is something at the end of my Ph.D. that I decided to follow, and I've been on that mission of trying to develop and commercialize algae biofuels ever since. I will get there in a minute and talk about that journey and some of the learnings I've had and reflect back. But again, I want to say it's great to be back here at MSU. It's my 3rd time back here, and fortunately, the last time I was back in 2022, I was on the other side of something that was very important to me when I was a student. As mentioned earlier, the Choices Day, where people outside of academia who are on their career path in different industry, government positions, tell their stories to students, and we get to ask our questions and learn more about these paths that are very mysterious to a PhD student at the time. It was great to be back on the other side of that. And that was about 12 years into my career at a company called Synthetic Genomics that had then rebranded as Viridose. Again, get into it in a minute. But had been entirely on the technical side and rose up through the ranks and was overseeing the science at the director level. And I remember very clearly at the time, what I told folks was, at some point in your career, you also learn about what it is that you don't want to do. And for me, it's all about staying closely connected to the technology, overseeing the science. This is what I love. And I know, for example, one thing I don't ever want to do, as far as I can tell, is become the CEO. And well, a year and a half later, 13 and a half years into the mission, became the CEO of the company at a time that was very challenging for a couple of different reasons. We had been very successful in continuing to drive the technology towards the path of the commercial thresholds that we need to really bring this technology to start scaling in a way that matters in this massive market of fuel, so that we can have a fuel that goes into the wings of planes that is not nearly as much an emitter of CO2 as we currently have, right? It's a difficult and intractable problem and I've loved being on that mission for years. Right now, we've gone through a major series of evolutionary changes, but I want to revise my statement to the graduate students, because I think there's still a few of them that are in the room. What I'd say now is never say never, guys. You don't exactly know where your path is going to take you. It's been an absolute rewarding, challenging experience to go from that position where I was in 2022 to where I am today. And on that front, I just want to talk about on mission. Going back to 2010 when I left PRL, what I joined was a company that was rapidly growing and building different business units. I joined at a time when the company that was called Synthetic Genomics, founded by Craig Venter and Hamilton Smith, both very different people in terms of their personality type, but very mission-driven. In the case of Ham, who I connected with more often, just really interesting and a humble advisor on his part. With Craig, kind of a different story with him in the CEO role. I won't talk too much about that today. But at any rate, I joined at the tail end of a huge hiring spree on this ExxonMobil-sponsored research program. I think my first title was scientist with lipid specialist in italics or something like that. And the first thing I did was not work on lipids at all for like a year and a half. I helped screen the entire strain selection. The program as it started out was a complete soup to nuts, finding the best commercial strains, starting to build the genetic tools to develop them, also building a greenhouse, outdoor waste ray ponds, and trying to get everything done, the downstream processing. What we realized about a year and a half in is that there are a lot of fundamentals that we need to focus on first, and there were some cool level heads that won the day in that. And what ended up happening was an over 13-year collaboration where we started out really focusing on the critical fundamental issues that needed to be solved before you could get to the tenfold improvements that algae strains need to be at that commercial threshold. And it really started out with building great science at the company in a couple of different areas, trait discovery platforms that we could find individual traits that provide genetic gain. We heard in the last talk the importance of trait stacking. One trait, you're not one trait away from commercialization with algae. We've taken a slightly different path there, but developing a very efficient trait stacking platform to get these genetic gains in and accelerate the process of driving performance where things need to be. A lot of that proof of concept work was done in early strains that weren't commercial. And so later on in 2015, 2017, the work began to get that in a true industrially robust strain that had the legs to grow outdoors. And in 2022, focusing on taking the strains with improvements in the lab, going out to scale at multiple acres, and demonstrating the genetic improvements that lead to performance improvements can actually scale outdoors. And so a lot of those accomplishments working with the team have been great. It happened in a backdrop of a company that was doing a lot of other things. And in 2023, or late 2022, there was a seismic shift at Exxon with respect to their business priorities. And the collaboration ended, but it ended within a runway that allowed us to go out and raise a Series A round. At that time, I became the CSO of the company. And really got lean and mean, put the strain engineering on hyperdrive, and just last summer, what happened is we ran into a cash crunch, and it became apparent that our only path forward was to do some substantial reorganization of the company that included a Chapter 11 bankruptcy, starting a new co, transferring the assets, the core team of 10, these awesome people that I've worked with over the years that were on the previous slide. It was pipettes down, working with a lot of lawyers, interfacing with a judge in Delaware, dealing with creditors who aren't too happy with me. A very big change, but all things that you can navigate along this path from, not really knowing everything you need to succeed, getting some Kitty Hawk moments behind the belt, getting to a point where a lot of the proof of concept work is there, but we've got a lot of work to do because the plane ride that I took here, didn't have a drop of our product, and if it did have any SAF in it at all, very little. There's a lot of work to do in this space to go if we're going to reduce the emissions of heavy transport fuels. What I will tell you is pipettes are back up in hand. The ponds have the green stuff in them again. Paddle wheels are turning. Wish us luck. Stay tuned. Look forward to telling you more about that in the future. All of this is to say, for me, on missions like this, missions like the PRL, and I think it was mentioned earlier by Doug Gage, part of the success factor through the challenges and stuff is the ability to evolve, be flexible, and learning how to tack in the headwinds. That's something that I started to learn largely through observation when I was here at the PRL. There's these 3-year events where you have to go through renewals, and there were a few ones that had more complexity to them during my tenure with some changes in direction of the PRL. Watching the leadership amongst the faculty work together on those problems, get the right solutions in place, and get the continuing funding was a real big learning factor that obviously going through something like this, there's different people that you need to work with. It's a different problem, but going through this, I've got a toddler at home. He watches Mrs. Rachel or Miss Rachel. It's a great show if you haven't seen it yet. But she's got this saying, you can do hard things. And I kept saying that to myself through the time, like every time something was getting a little crazy with this process, the bankruptcy, it's like, no, you can do hard things. You can do hard things, and you can. So I wanted to come back to learnings at MSU. First one, and there's a ton of them. I just picked 2. The first one, back in the day, the technology was starting to move real fast in sequencing, and how is it that you're going to find your mutant of interest once you've got that mutant? A lot of what we were doing at the time was this, a set of really elegant approaches. This one I'm showing Cosmin mapping, which I don't know if there's that much activity in that space. There's probably still map-based cloning going on. But at the time, this was one approach, and I had done some of this, but there were a number of other colleagues working on this mysterious gene FAD4. And what was interesting about it was, there were some folks in a postdoc in Christoph's lab, Jianping Hu, and then Amada Jawi in Rob Last's lab. This was a collaborative project. And if I remember correctly, it turned out the original mapping back in the 80s was on the wrong chromosome. And some more recent mapping said, okay, it's within a couple hundred kb on this other chromosome. Well, the next steps the traditional way would have been keep doing the mapping. I think what happened, I'm pretty sure, because I was part of the discussions more about the future of what could happen, was within a couple hundred kb, it's like, okay, here's the genes from the genome. Let's sequence the ones that look most likely, and then bang. And what we were talking about at the time, these babies didn't exist. But what we were talking about at the time was, because 454 was around, Illumina, you were starting to see transcriptomics data sets popping up. It was like, what if you could burn the haystack in like a week and just get that gene? And that in the early days of my career in industry was a big focus, getting there. And so technology moves fast. You got to stay with it. The second one I'll just mention real briefly, just for my love of TLC plates. At the time in Christoph's lab, we were very TLC plate centric. And every once in a while, one would say, well, why don't we just switch over to mass spec? And he had a good rationale about us. And I won't go through it all. He can share it with you if he wants. But basically, over the years, in reflecting on it, it was like, this was a great discovery tool. Yes, it wasn't the most sophisticated in the world, but in an afternoon, you could extract your lipids, you could separate them out, you could cut them out, run GC, and answer your questions. While in the mass spec, sometimes you might be working on that for a couple months just to develop the platform. Over the years, at SGI, Viridis, Nuco, there have probably been a half dozen times where I've been the hero of the day by pulling out a TLC tank, isolating something, whether it's a pigment or a lipid, while the analytical chemistry team is talking about a couple $50,000 project to get things booted up on a mass spec. And so one of the things that was really cool for me in this difficult time of packing up the lab was the team, the core team that's been with me for years, that stayed along and helped pack up the lab, they already knew the TLC plates were going along and had packed them up, and they just said, yeah, the TLC plates are labeled, they're going into storage from now, because with TLC, never say never, for me anyway. Mass spec's also packed up too. We're gonna bring that back online pretty soon. But what I've learned from this is obviously with technology, it moves so fast, keep up. But in the world that I moved into, I found it real important to have this principle around finding the balance between the cutting edge and the hilt when your job is more about building a stable technology platform that has to be robust across time, cost effective, and not have too many Maseratis that break down every other week, so to speak. And so that's the principle that I started to learn about at MSU. How am I doing on time? I tend to just blow time out of the water. I don't.
So you have another 10 minutes.
10? Oh, wow. I'll slow down and take my time on this one. This story on science goes back to this white whale, GGGT, that the biochemistry had been solved or proven out in, I think, the early 70s, right? And was originally believed to be the major enzyme involved in galactolipid biosynthesis. See, I can't say galactolipids anymore. And there'd been no traction on figuring out what the gene was and what the actual role was for this gene. And so, it was probably second year, third year, I'd started thinking about, well, how to develop a screen. What's a clever screen where we could trap this gene? And came up with a strategy where we would take the DGD1 mutant that had been suppressed by TGD1, I believe. We would mutagenize that plant and then look on TLC plates, which is kind of a brute force method, but after a couple of years, you got really good at loading up a TLC plate with 40 spots and running 80, 100 a day. We found one, but this damn plant, after a couple months, was doing nothing but spitting out weird little rosette leaves. And I read the literature, and it's like, that looks exactly like the GA1 mutant, so I'm gonna spray it with some GA. And it killed the thing like within 2 days. And so I went into Christoph's office and I told him, and he was cool about it. He's like, well, you got the M2 population, right? And then he said, and you've got a lot of progress on other things, so let's hold this one off. And he had a good way, especially for someone like me in graduate school, without just saying, stop that, don't do that. Like, you've got enough going on. Like, put this one away for a while. What he didn't remember at the time was about a month before I killed this plant, was that he had sent me an email of a paper. Myself and Changcheng Xu in the lab, someone who I worked with very closely, was just in sync with for the entire time that we overlapped. And for whatever reason, Changcheng ignored the email. I read this paper, and Christoph said, hey, it was basically a one-liner in the subject line was, do you think that this could be the GGGT? And I read the paper, and it just clicked because I had read a dissertation of Wayne Rykoff, who had left right before me, and he had a chapter on the GGGT and a little section on how glycosyl hydrolases can sometimes act as glycosyl transferases. And it just connected with me like, yeah, it's on the outer envelope where the GGGT should be. I'm gonna order the tDNA seeds. In about 3 weeks or so, I don't know exact timing, but it was not too long after that sad story, I was able to walk in to Christoph's lab with a TLC plate that was still warm and had that alpha naphthol smell on it, that sweet smell that I kind of miss, and kind of flip it around and say, we got the GGT. He's like, what are you talking about? I was like, you sent me a paper like 2 months ago. I ordered the tDNA lines. And so he's looking at the plate, and he's like, what's this one? Wild type. What's this one? Wild type with the treatment that creates TGDG. What are these two? These are the knockouts in this gene SFR2 he's like, okay, they don't make it. And it's funny because Christoph and I, we're like, both of us are similar in some ways, different in other ways. One way that we're similar, like we don't really get up and do this kind of a thing, but he was like doing a weird pigeon thing, and I was like doing a chicken. Like we were excited, but we didn't really know how to show it. That was a fun day. What was also fun was setting up a meeting with Mike Tomashow's lab and getting really solid advice, like how do you responsibly mimic freezing in a lab? You know, the first idea was throw it in the -80 real fast. No, so it was a great way to kind of reconnect to this initial thing that brought me here, like in the last year. The data collection to kind of round out the story and build a hypothesis on why this, why this had evolved, why it's important, was really straightforward and a joy with folks. Bhagya, who is a master's technician in the lab, was a co-author on this paper. Really fun time, but what I go to today and remembering everything, yeah, it's nice to have a science paper land 2 weeks before your dissertation defense. But what's really nice is remembering that day. Like that was one of the funnest days. And growing into a position over the years where I'm overseeing science and less connected to it, trying to create those memories with folks is just as important as meeting technical deliverables. So the advice here, read the literature and have good advisors. Beware of repressor-suppressor screens. Maybe that's just a personal thing. Maybe it's not good advice. I don't know. So where to go from there? I didn't know if I was going to go way over, so this is just kind of the speed round. The big thing I think that I took away from the PRL outside of the science was, how to start taking what I call tough love or radical candor or constructive criticism, whatever you want to call it, from the best advisors that you can find, and also how to learn how to give it to folks. The things that we work on in the lab where you don't know what the answer is to something. Budgets are tight, timelines are short, especially in industry. You've got to meet technical deliverables. You can't do that stuff if people aren't capable of coming together, the best ones that you can find, and really collaborating well together. That's something that really stuck with me from my years at the PRL and work in Christoph's lab. And if you can't do it, you can't do great science. And great science is essential to make impact in this world. As all professors know, it takes a lot more than great science. It takes a lot more than great science to be able to build an ecosystem to do great science. That's one of the things that I also learned here just through observation. What does it take to be able to do great science? Learning other things along the line, working with lawyers. I've kind of had enough of that for the past couple months. I could give them a rest. But learning that it's important to sync great science with other areas was something that I got here. I got my start on it, and it's helped me a lot in my career. The last thing, because you have to do all those extra things, you can't. My initial thinking and why I like science so much was you just go into a lab and you work for a week. You talk to your boss once a week about the results, and then you go back. That doesn't last forever, right? And that's all of this stuff is part of why, worthy missions are hard, right? And what I tell people, especially people in undergraduate when I meet them, just be sure you join a worthy mission. The good news here for everyone today that's been a part of PRL, the PRL's past, the folks that are here today that are going to move the mission forward, you can already check that box. 60's young. Hope I get back. I'll be 60 before I know it. Time flies. That's my story. Sticking to it, and thank you.
Wonderful. I didn't know about the chicken dance. That's really any questions for Eric? Yes, jump in.
Just curious, why did Jay kill that mutant?
I think I put way too much chili on that one. I probably didn't read the paper enough and actually go across and talk to some of the great biologists of is it this or is it this. I think there was a little bit of emotion and I put way too much on it. And it was probably the detergent to get the GA into the solution that did it. That was 17 years ago, I don't know. But I do know I killed it because that wasn't the intent.
So you didn't go back to the M2? You just let it?
That was like that was the beginning of my 4th year.
Oh, yeah.
You know Christoph, he's like, wrap it up. And
I can hear saying that. Okay. Well, thank you so much. Please join me in thanking Eric for a wonderful talk.